recording. Hi everyone, happy week 7. Hope the first round of midterms went well. We are excited to do a journal club on Dr. Zeiger's research. He'll be coming in, I believe, week 9 to talk about his career in MD, PhD degree, which is very cool. Just a quarter overview for today. Week 8, we have Outreach hosting our GM. Excited for that. Week 9, as I mentioned, we have Dr. William Zeiger from the UCLA Movement Disorders program coming to talk to us. And then week 10, I believe we have a tentative in-person social being planned right now. So look out for info on that. All right, and for today we're bringing back our icebreaker questions. You can choose one or choose all of them. What is the best spot on the hill / campus to study? If you could create and teach your own class, what would it be? And what is your guilty pleasure activity? All right, I'll shuffle the breakout rooms. Let me see. Okay, 4 is a good number. Okay, great. We'll spend 3 minutes on icebreakers and come back to the journal club. All righty. Feel free to share any, special answers in the chat. But we're going to move on to today's presentation. All right, I'll pass it on to education.
Yeah, so hey everyone. Today we're going to be talking a little bit about what Dr. Zeiger does at UCLA and give you a background on the work that he does, and then also do a short journal club on one of his papers that he released, just so that you guys have an idea of what he'll be talking about next week and potentially have questions that you can ask. So we're just going to go right ahead and start with neural circuits. So next slide. Oh, I think this is, sorry. So yeah, so neural circuits are basically just a population of neurons interconnected by synapses to carry out a specific function when activated. And so basically they interconnect to form large scale brain networks, and they're categorized into 4 different types. So a diverging circuit is basically when one neuron synapses with a number of postsynaptic cells, and then each of these may synapse with many more, making it possible for one neuron to stimulate up to thousands of cells. So like, for example, neuron A is synapsing onto neurons B and C, so that'd be diverging. Converging circuit is when there's inputs from many sources that are converged into one output. So neurons B and C will synapse onto neuron D, that's a converging circuit. A reverberating circuit produces a repetitive output. So in a signaling procedure from one neuron to another in like a linear sequence, one of the neurons sends a signal back to the initiating neuron, so kind of like a feedback loop. And each time that first neuron fires, the other neuron further down the sequence will fire again, sending it back to the source. So for example, neuron A fires onto B, and then neuron B will fire onto C, but then C will fire back onto A. And so it'll provide like a circular feedback loop. Usually most of these are kind of like more like a negative feedback loop. So if there's too much information going one way, the last neuron that's being synapsed on can decrease some of that input, so there's not too much going on. The parallel after discharge circuit is when a neuron inputs to several chains of neurons, and so each chain is made up of a different number of neurons, but their signals converge onto one output neuron. And so each synapse in the circuit will act to delay the signal by about 0.5 milliseconds, so that the more synapses there are will produce a longer delay to the output neuron. So you can kind of think of it as kind of like a diverging circuit and a converging circuit, both connecting to make one whole circuit. So it requires the both of them. And then the time delay just allows for a little bit more synchronization so that the cells don't get overpowered. Next slide.
So next we're going to go over diverging and converging circuits in detail. So divergence is basically a mechanism that allows one neuron to communicate with many other neurons in a network. which means that individual neurons can connect to many different postsynaptic cells. And postsynaptic cells are just neurons receiving a signal, and they can receive signal from multiple presynaptic neurons. And convergence is just another mechanism that allows a neuron to receive input from many neurons in a network. And a single postsynaptic cell receives input from a number of different presynaptic cells. And presynaptic cells is basically just neurons sending the signal, and they can contact multiple postsynaptic neurons. So the basic rule of thumb for divergence and convergence is that convergence is one input leading to many outputs, and convergence is many inputs leading to one output. And next slide, please. Okay, so before I explain an example of a neural circuit, I would like to ask a fill-in-the-blank question. So does anyone recall what are the 3 types of neurons? Feel free to share out by unmuting, or you can type in the chat. Anyone remember what are the neurons? Yep, I see motor neuron. And sensory neurons is also correct. There's one more, and the picture there might give a hint. Yes, interneurons is the correct answer. So the 3 types of neurons again is sensory neurons, interneurons, and motor neurons. And they're all going to play a role in the stretch reflex that I'm about to talk about. And so the stretch reflex is basically, so like if you been to a body check or something, you probably experienced this. Your doctor will tap your knee. And so the sensory neurons on the knee sends an electrical signal to the brain, and that activates the interneurons. And this in turn activates the motor neuron. And then the motor neuron will send a signal back to the muscle that results in an extension of the leg. So this is one simple example of a neural circuit. And convergence and divergence is necessary for the circuit to successfully operate in real conditions. Next slide, please. In detail, divergence is when the doctor taps your knee, a single sensory neuron is signaled, and it diverges this synaptic information into various synaptic connections with many individual motor neurons. And this causes the leg muscle to contract. And that convergence is when the muscle is stretched. It is because multiple sensory neurons converge into one single extensor motor neuron, and this allows the leg to extend.
So our next section here is going to be on neuroplasticity and how all of these neural circuits play a role in like development of these circuits. And Tiffany, you can play the video, please. Oh, Tiffany, there's no audio. I think you need to share computer audio.
Hold on 1 second. All right. we go. Let me know if this works.
Not so long ago, many scientists believed that the brain did not change after childhood, that it was hardwired and fixed by the time we became adults. But recent advances in only the last decade now tell us that this is simply not true. The brain can and does change throughout our lives. It is adaptable, like plastic. Hence, neuroscientists call this neuroplasticity. How does neuroplasticity work? If you think of your brain as a dynamic, connected power grid, there are billions of pathways or roads lighting up every time you think, feel, or do something. Some of these roads are well-travelled. These are our habits, our established ways of thinking, feeling, and doing. Every time we think in a certain way, practice a particular task, or feel a specific emotion, we strengthen this road. It becomes easier for our brains to travel this pathway. Say we think about something differently, learn a new task, or choose a different emotion. We start carving out a new road. If we keep travelling that road, our brains begin to use this pathway more, and this new way of thinking, feeling, or doing becomes second nature. The old pathway gets used less and weakens. This process of rewiring your brain by forming new connections and weakening old ones is neuroplasticity in action. The good news is that we all have the ability to learn and change by rewiring our brains. If you have ever changed a bad habit or thought about something differently, you have carved a new pathway in your brain and experienced neuroplasticity firsthand. With repeated and directed attention towards your desired change, you can rewire your brain.
I hope that video was a little helpful for you guys to understand what neuroplasticity is a little bit more. But basically, neuroplasticity is when the brain basically changes its connections in order to adapt to the environment, adapt to changes in development. It's basically the idea that the brain is not static and is able to respond to changes. And yeah, it would relate to, I guess it could relate to the growth mindset because you're able to form new synaptic connections if you, I guess, change studying methods, for example. Yeah, so the brain strengthens, prunes, and forms new synaptic connections. There's what's called long-term potentiation. An AMPA receptor is basically a receptor that is used for glutamate and like other cation channels, and this can lead to increases in synaptic connections. So when a synapse becomes more excitable, it's correlated to like an increase in the strength of that synapse. So that's also what we can call like learning. We're able to form new connections over time when we strengthen those. There's also long-term depression. So if a synapse isn't used or a particular neural circuit isn't used, that becomes a lot weaker over time. And then eventually there may be, that connection may decline and we'll see a much smaller response. So structural changes are constantly occurring in the brain. And so this is why we see increases and decreases in synapse size and number and density over lifespan. Oh, next slide.
Yeah, so there's 3 types of neuroplasticity. So the first is experience independent. So this is mainly involved with like prenatal developmental phases, and basically involves brain changes that take place regardless of the environment. And this makes the most sense as to why it would happen prenatally, because these are kind of your fundamental synapse growth. And so you want to make sure that evolutionarily, we want to make sure that every child has the same neural pathways. And so that's why a lot of these don't require an environment to induce certain changes. For the experience expectant one, these are also independent of external factors, but mainly they assist neurons in connecting to each other independent of other processes. So just making sure that there's connections in between major parts of your brain and also your spinal cord. And so an example would be the retinal ganglion, which involves axons coming from the retina and connecting to other parts of your brain and your eye structures. experience dependent is dependent on the experience. So it will change in response to different situations. For example, we're talking about growth mindset. So like Sasha said, if you're changing, for your learning habits, or you associate different things with your studying, that will lead to changes in your synapses, potentially making it easier to learn a certain new topic. So other examples include moving to new territories, learning from problems, or suffering from injuries. And so it's related to challenges that increase or decrease synapse numbers. So anytime you're in a different environment, your synapses will change to help you adapt better to that environment. Next slide. And so now we're going to transition into talking about more specifically about Zeiger and what he does in his lab.
So one main focus of the Zeiger lab is studying ischemic stroke. So it's one of the more common of the two types of stroke. So I already said one of them is ischemic. So does anyone know what the other type of stroke is called? It starts with an H, if that helps at all. And you can put it in the chat if you don't want to unmute. Allison got it. It's the hemorrhagic stroke, and then the other type is ischemic, which we'll be talking about. And an ischemic stroke is basically when blood supply to part of the brain is interrupted or reduced due to either like clotting or narrowing of the artery. And because of this, like you don't have as much blood flow to the brain. So then oxygen and nutrients that are in that blood fail to reach the brain tissue. And it can lead to death of brain cells in a really short period of time. So if you look at the picture down below, the left-hand side would be like what a normal brain would look like. And then the right-hand side would be if you were going through an ischemic stroke. So you can kind of see there's a lot less like blood flow to that area of the brain. And it's kind of really visible in the scan. Next slide.
So the Zeiger lab uses the Photothrombotic Stroke model. So if you guys took a look at the journal that we have for this week, you'll notice that they don't necessarily use ischemic stroke, but they'll have to instead induce stroke in an animal model. So the Photothrombotic Stroke model, what it does is it simplifies the study of ischemic stroke, especially because ischemic stroke has a pretty short therapeutic window. It can be very difficult to study in a non-controlled setting. So what the photothrombotic stroke does is the aiming of light towards a particular region can result in the occlusion of arteries. So you'll see a much larger region where there's a lesion, and that can be studied more in depth for like therapeutic purposes or to learn more about how circuits are formed. So what the lab's goal is with this stroke model is to see how these neural circuits are either able to be rehabilitated or whether other neural circuits move to take their place. Oh, next slide. Okay, so here we're gonna do an overview of calcium imaging, which is the technique that's used to detect how these neural circuits either remap or don't. And this is one of the methods that were also mentioned within the journal. Because the lab's interested in circuit dysfunction, calcium imaging can be really useful. But before doing that, we just want to quickly review what does calcium influx do at the axon terminal. For the first part, there are two words, and the second part, it's either endocytosis or exocytosis. So those are two, that's like a very big hint. But yeah, if anyone wants to like put it in the chat or unmute themselves, feel free. Does anyone remember what happens at the axon terminal after the calcium influx? Hit LS7C, Tiffany's right. Does anyone remember? And even if not, totally okay. Yeah, Max, you got it right. It says neurotransmitter release into the synaptic cleft via exocytosis. So what calcium influx at the axon terminal does is it leads to neurotransmitter release at the synapse. And these are exocytosed as the vesicles fuse with the membrane. The reason why this is so important is because it makes calcium a really important and easy ion to tag when we're trying to understand neural circuits. So specifically in this paper, we're using calcium as a way to visualize whether a circuit is functioning or not. And there are fluorescent molecules that are bound to these calcium ions. And that way, under a microscope, you're able to tell where the calcium is moving. And there, next we have like a video to explain this a little more.
Modern recombinant DNA technologies and microscopy can be combined to modulate and visualize molecular interactions. As an example, fluorescent calcium imaging can be used not only to observe neuronal activity, but also to associate behavioral characterizations with physiological states. In our research, we use calcium imaging via recombinant DNA technology and microscopy to not only observe neuronal activity, but also to associate behavioral characterizations with physiological states. In order to detect the movement of calcium ions during neuronal signaling, a genetically engineered calcium indicator, or GCAMP, is required. GCAMP is a hybrid calcium sensor encoded by the calcium-binding messenger protein calmodulin, the calcium N-calmodulin binding M13 peptide of myosin kinase, and the fluorescent protein GFP. Few interactions are maintained between the GCAMP protein domains, and as a result, GFP fluoresces minimally in the absence of calcium. In the presence of calcium, calmodulin undergoes a conformational change and is free to bind the M13 domain. This induces GFP to adopt optimal conformational changes for increased fluorescence. The increased fluorescence can then be detected visually under a microscope. Let's see how we can visualize these molecular changes at the cellular level. We can inject model organisms such as the nematode C. elegans with plasmids that contain genetic sequences that will give rise to our calcium sensor protein. As a result, we can observe the relative changes in calcium levels in the cells of an organism over time. Calcium signaling plays an essential role in neuronal signal transduction.
Oh, you can go back one slide. Yeah. Okay, we're good, Tiffany. Yeah, so today's journal club is going to be on Dr. Zeiger's most recent paper. I know that the title is a little bit of a mouthful, so I will explain a little bit of like what some of these terms might mean, because I know, yeah, they're pretty new. They were new to me too, so I think explaining the definitions will help. But the barrel cortex is basically a region of the somatosensory cortex that's pretty unique to rodents, and this is because there are anatomical units called barrels, and these each correspond with like a whisker on these rodents because they use whiskers to sense get information from the environment. So that's specifically what this is referring to. The experiment uses, I don't remember whether it's mice or rats specifically, but that's the region of the brain that they're exploring. And this word potentiating responses, you can just view it as like rehabilitation. So what it's saying is that neuroplasticity is reliant on the rehabilitation of neural circuits and not remapping of the new circuits. That's it. I know, because I was confused by the terms like barrel cortex and potentiating responses. Yeah, I hope that makes a little more sense before we get into the actual activity. Oh yeah, Tiffany, if you could create the breakout rooms, that'd be great. So we have one abstract group, one introduction, and two discussion. And oh yeah, please make sure when you're discussing in your rooms to come back with some takeaways from your sections so that we can have a discussion after, and get people assigned to share out beforehand so that, you know, we get to hear all of your ideas from the rooms.
All right, all the rooms have been opened, and the link to the paper is in the Slack chat.